I'm going to introduce our first speakers. This segment overall is going to be led by Dr. Hans Bjornsson of Johns Hopkins University and of University of Iceland, along with his team. And as Pat mentioned earlier, we had the pleasure of meeting Dr. Bjornsson originally about 5 years ago. And it was in large part because of discussions with him, along with Dr. Harris at the time, that really became the spark for us creating this foundation. So we're really pleased to have him kick off the conference today. Dr. Bjornsson is going to speak to a bit of the research landscape for Kabuki syndrome and really how that's changed over the last 10 years since gene discovery. So with that, we're going to actually start with a brief video that relates to Dr. Bjornsson's program and his team. So I think we've got that ready to go, and then I will turn it over to Dr. Bjornsson.
The main mission of the Bjornsson Lab is to try to understand the role of epigenetics in disease through the study of the Mendelian disorders of the epigenetic machinery. Kabuki syndrome is a prototypical Mendelian disorder of the epigenetic machinery. Individuals that have Kabuki syndrome have growth retardation, immune problems, and intellectual disability. The dogma for intellectual disability has kind of been that intellectual disability is something that you can't treat after birth. We think that our work with work from other individuals suggests that this dogma may not be true, that at least for Kabuki syndrome, it may be a treatable cause of intellectual disability. Our lab has been able to have a sustained focus on Kabuki syndrome and also come up with some therapeutic strategies for Kabuki syndrome and related disorders.
So I'm absolutely thrilled to have a chance to speak today, and I continue to feel privileged to be part of the Kabuki community, and just really enjoy this, seeing the amazing progress that has happened. Okay, I was tasked with giving insights. So the Kabuki syndrome gene was discovered about 10 years ago. And I'm mainly going to talk about our work, but I just want everyone to understand how quickly things have moved for Kabuki syndrome. And I just want to start with some very basic things, just so everyone kind of keeps track of the whole conference. So what is DNA? DNA is the language of life, how cells know what to do. And you could think about the entire content of the DNA for an individual as the book of life, the human genome. And if the human genome were a book, the genes would be words. And we've had this book now for 20 years, but we still don't quite know how to read it. And the reason for this is this same text, this book of life, needs to be read in at least 300 different ways. And the reason why I say that is that there's at least 300 different cell types, and some of them are quite different. If you think about a sperm versus a neuron, they have very different components. So how could one theoretically achieve this? So one way to read a book in different ways is to highlight out the specific genes that you need for that book, and maybe have a separate highlighting system for every cell type. But how is that actually done in cells? And I'd argue that one way to kind of conceptualize the epigenetic machinery is to think about it as the genome's highlighting system. So what are these epigenetic marks? These are modifications of DNA or the associated proteins, and they're heritable through cell division. They're reversible and affected by the environment. And if nothing else, they have the potential of adding to the information content of the DNA. And there are two big groups. There's DNA methylation, and then there's the histone tail modifications, which is what I'm going to introduce today. So DNA is not floating around in cells in the linear state. It's very heavily packaged. And this is kind of the basic unit of packaging, something called the nucleosome. So DNA is wrapped around proteins that are called histones, and they have these tails that stick out, and those tails can be modified, and that's what's called histone modification. And chromatin kind of exists as open or closed. And I just want you to know that there are certain modifications that are seen in open, like H3K4 trimethylation, and others that are seen in closed, like H3K27 trimethylation. What are these weird names? H3 what? So here I'm showing you one of those nucleosomes, and I'm showing you the two tails, the H3 tail and the H4 tail. And what H3K4 means, it just means it's the fourth lysine of this tail. Same as H3K27, it's just the 27th lysine on the H3 tail. And these are the two modifications that we think are relevant for Kabuki syndrome. So just to summarize here, the epigenetic machinery is a highlighting system of sorts that regulates other genes through the additions of these chemical modifications called chromatin modifications. And there are many of these, but I just want you to know that there are certain marks that we see in open chromatin and others that we see in closed chromatin. So what then happens when the epigenetic modifications or machinery are disrupted? So it turns out that Kabuki, the two types of Kabuki, I put a little heart next to them, they're part of this emerging large group called the Mendelian disorders of the epigenetic machinery. And there's about 70 genes. And in this big pizza pie, I'm showing you each gene as a slice. And the two Kabuki genes are up here on the top. And there's about 23 writers, 10 eraser disorders, 27 reader disorders, and 10 remodeler disorders. And by looking at the entire group, there's some power in that because it teaches us some lessons. And one lesson that we can see is that for this entire group, if you look at the blue color, this is the conditions that lead to intellectual disability. So all these kids have intellectual disabilities. That's a unifying feature of this whole group. Many of these are enzymes, and in this group, it turns out that they are haploinsufficient. They're dosage sensitive, which is quite unusual for enzymes. And then the final point, and you'll hear more about this by Leandros, is that we think that all of these factors lead to epigenetic dysregulation. And there's now a large number of these, and they don't work in isolation. They actually collaborate. So you'll hear about other disorders than Kabuki, but they're kind of working at related marks that have overlapping roles to some extent. And for the rest of the talk, I'm going to focus on Kabuki. So Kabuki is this disorder of histone methylation. There are two different causes. One is dominant, the other one is X-linked. And the two genes are called KMT2D and KDM6A. And all the patients have these unusual facial appearances, so that many of them look quite similar, like sometimes like their siblings or related. But they all have intellectual disability. Many of them have immune problems, and many have growth retardation that happens postnatally. And the first one that was discovered only 10 years ago, just to tell you how amazing progress we've made, was called KMT2D and puts on this open chromatin mark called H3K4 methylation. 2 years later, another group found KDM6A. It removes this closed chromatin mark, H3K27 methylation, and both of these affect the ability to open up chromatin. So I, as a physician scientist, always looking for therapeutic potential, I had this relatively naive idea. My idea was that maybe at some genes, the balance between open and closed chromatin is very tightly maintained. And maybe in Kabuki syndrome, the balance is tilted towards closed chromatin, so they cannot open the chromatin of these genes that they need. And if that were true, we could maybe use drugs that open up chromatin to treat aspects of the phenotype. And to do this, we had to characterize one of the first mouse models of Kabuki syndrome. We call it the beta geo mouse or the Kabuki mouse. And it turns out that this mouse has many features that are similar to what we see in patients. Blue are the average mice, yellow are the ones that have the Kabuki mutation. And you can see they're smaller after birth. And they have the same immunological profile as our patients do. They have low IgA levels. So that's good. It seems to be reflecting what we see in patients. We've also found, in collaboration with other groups, we've found a couple of new phenotypes that we think could give insights into mechanism. The first is we see an expansion of the growth plate, which was very surprising because these patients and the mice have shorter bones. And this was done with Jill Farner at Johns Hopkins. And then with Andrew Lindsley at Cincinnati, we discovered that the mice seem to have smaller and fewer Peyer's patches, which are these immune controlling centers of the gut. So that's also very interesting and can lead us towards some mechanism. But my biggest interest was the neurological phenotype, because there had been this dogma that you heard about in the video that you couldn't treat intellectual disability. And we wanted to see if we could measure the neurological dysfunction with an assay. We started out with something called novel object recognition. So here you put a mouse in a cage, you put two little objects, the mouse can freely explore them, and then you switch out one of those objects for a new object. And a normal mouse should spend more time with a new object because they remember the old object. And what happened is that there was no difference between the average mice and the ones with the Kabuki mutation in the first phase. But in the second phase, what we saw is that the Kabuki mice spent less time with the new object. So that pointed that there may be an hippocampal memory defect. So we went to a more robust assay, something that's called a Morris water maze, and it's a tank filled with water. We take a full week to teach the mice to find an invisible platform. And then the last day we remove that platform. And what we found is the Kabuki mice in yellow crossed about half as often as the average, the other mice. So they seem to truly have a hippocampal memory defect. The other thing that we did with these early studies, at that time, people were mainly focusing on H3K4 trimethylation. It's a little bit changed since, but we did this test called ChIP-seq to look at the global distribution of this mark. And what we found is that there was a defect in the Kabuki mice compared to the other mice. However, when we put the mice on a drug called AR42, which opens up chromatin, we could rescue this defect. So a summary here is that two types of Kabuki syndrome suggest that maybe an imbalance between an open and closed chromatin state plays a role. And our mouse model seems to reflect the features that we expect for Kabuki syndrome, but they also have this hippocampal memory defect that we can measure. And finally, they have a biochemical abnormality that we can rescue. So we next wanted to look at the brain, and we noticed that there was an area of the brain called the granule cell layer, the dentate gyrus, that seemed to have a lot of KMT2D. And this is part of the dentate gyrus, part of the hippocampus. And it's a very amazing place because it's one of the few places where you keep making neurons throughout your life. And it happens kind of at the inner arms of this V-shaped structure here. And if you disrupt this process, you get hippocampal memory defects, which is what we see in our mice. And one of the students early on started pointing out and saying, hey, it looks like the layer is thinner in the Kabuki mice compared to the others. So we quantified that and we could show it was in fact thinner. So then the question was, could there maybe be a problem in making new neurons in the Kabuki mice? So here we've stained against something called double cortin. And you can see in the average litter mates, there's a lot of these double cortin positive neurons. But in an age-matched Kabuki mouse, there's fewer of these. And this we've quantified many times, and we see it again and again and again. So then the question, is this something that we can manipulate? Can we change this in our mice? And here we've treated a full cohort of mice with AR42, and we're doing a dose curve. So these are the wild types on top, the Kabukis on the bottom. And you can see there's a big difference in the vehicle. They got no drug. You can see there's more green cells in the average, the siblings compared to the ones that have the Kabuki mutation. But when we increase the drug, we can rescue this. And we've done this many times now. You can see there's a dose-dependent rescue. As we increase the dose of AR42, the Kabuki mice get closer to what we see in the other mice. What about the behavior? Can we fix that as well? So it turns out when we do this test, the platform crossing in the Morris water maze, The Kabukis cross about half as often as the other mice, but on 10 milligrams per kilo per day of AR42, they both did a little bit better, but now the Kabukis caught up to the other mice. So the summary here, a mouse model of Kabuki syndrome reveals this thinning of the granule cell layer of the dentate gyrus, which we think is caused by impaired neurogenesis. And I just want to point out how rapidly things happen after the discovery of the gene. Only 4 years after the discovery of the gene, we had this proof of principle study to show that the defects in the dentate gyrus could be reversed using drugs that target the epigenetic machinery. And that kind of suggests that at least aspects of the phenotype can be treatable after birth. And amazing stuff has happened in the decades since the gene was discovered. For one, and this is not a great metric because it's just one metric, but if I just look up Kabuki syndrome in the title of papers, the numbers of papers per year has doubled. That's real progress. And it's impossible in this short time to cover all the advances. Thankfully, a lot of people are going to be presenting during this conference to show you what's new and so forth. But I just wanted to touch on 2 things. One is, has our work been replicated? You know, some of this is in mice. How does it reflect on patients? And the first replication came from France. A group there decided to ask the question, is the dentate gyrus smaller in Kabuki compared to controls? And the answer was yes. So that replicated very nicely. Other piece of replication, which you'll hear more from Jackie Harris, she had this beautiful idea that if truly adult neurogenesis was disrupted in Kabuki syndrome, they should have a worse ability to perform in visual spatial tasks like block design. And when we did the study, in fact, that's exactly what we found. Block design seemed to be the task that Kabuki individuals struggle the most with. The other tasks that don't have to do with visual spatial discrimination did better. So that's another replication in the patients. But there's been enormous progress in the last 10 years. There's been multiple people replicating aspects of our work or finding new agents. Some of it has come from our group, but some of it has come from other groups where people have shown that epigenetic agents can affect either the in vivo or in vitro cellular phenotype. But there's also been these amazing other strategies, you know, from read-through, which is a classical way by Giuseppe Merla, ERK inhibition, and Notch signaling inhibition as well. And you'll hear some of these stories during this conference. So what do I think are the most important things going forward in the next 10 years? I think we really need to nail down the mechanistic basis of Kabuki syndrome. If we truly understand the mechanism, we'll be able to develop the best therapies with the least amount of side effects. And you'll hear a little bit of insight from Leandros after my talk on this aspect. We also need to broaden our models. We need, you know, the Kabuki mice is really just one genotype. We need even in mice, we need multiple mutations so that we truly understand Kabuki syndrome. And we need robust outcome measures. We need something to measure during a clinical trial. And you'll hear more about that by Jackie Harris and Alison Kalinowski. And finally, we need to establish the window of opportunity. When can we implement this treatment? Does it have to happen at birth, before birth, after birth? And you'll hear stuff by Hadia just after my talk about this idea. And finally, I'm running out of time, but I just want to say, how can we work together? Participate in research, not just with me, with anyone that wants to work on Kabuki. Organize meetings like this one. Fundraise a small amount of money can really make a big difference, and then keep lobbying for Kabuki syndrome. These are the amazing people I work with. 2 of them now have their own labs, Jackie Harris and Jill Farner. And I just want to acknowledge my funding. This couldn't have been possible without the Luma G Foundation, which has allowed me to have a sustained focus on Kabuki syndrome and has allowed me to get funding for Kabuki from the Icelandic Research Fund. And I've also been supported by many Kabuki families. I just want to mention the Benjamin family, but a lot of families have helped me move the science forward. So thank you so much.